うん。 Mm-hmm. Okay. Oh, what just happened? Okay. I don't even remember what's in this one.
They don't just digest lipids, they actually build lipids. So they resynthesize triglycerides, they put the fatty acids back on the glycerols, but in the way that they want to build them. Okay? So now you've rebuilt these lipids inside the cell. So it's intracellular activity going on.
Okay, I think I fell asleep when I was watching this. Okay.
Now, once we look closer at that brush border of microvilli, it became gradually obvious. At first we thought it was just increasing the surface area, which it is. But in the lining of those cells, there are actually enzymes involved. And those enzymes are doing quite a bit of the final digestion. It's actually happening in the membrane of the cells. As the food is absorbed, it's being digested. And so we call these the brush border enzymes. Brush border enzymes. The enzymes that are bound to the cells lining the intestine. Okay, so digestion in the small intestine is going to use these pancreatic enzymes and these brush border enzymes. Okay, so let's sort of summarize the digestion of macronutrients here. That's for carbohydrates. Carbohydrates, of course, are chains of sugars. So here's glycogen, that's the way animals store carbohydrates. Here's cellulose, what plant cell walls are made out of. Here's starch, the way plants store carbohydrates earlier. Okay? Now we know that no animal can digest cellulose. So any cellulose you digest, or that you ingest, is not going to be digested. That's what we call fiber. That's insoluble fiber. So how do we get the digestible carbohydrates digested? Well,
Oh wow, wait, termites don't actually eat the wood. That's crazy, bro. Arthro means jointed, right?
first step is the salivary and pancreatic amylases. So salivary amylases, we already said, probably don't do much because we don't need food in there long enough. Pancreatic amylases are breaking things down into disaccharides and trisaccharides. So we have, if both glycogen and starch are chains of glucose, then if we break it down into 3 glucoses, that's maltriose. If we break it down into 2, it's maltose. So the pancreatic amylase in the small intestine is breaking these glycogen and starch molecules down into some free glucose, some maltose, and some maltriose. Now, additionally, we may consume other disaccharides. So table sugar, make it like a spoonful of sugar, is sucrose. So that's a glucose and fructose linked together instead of 2 glucoses. Or lactose, when you drink milk, is a glucose and a galactose linked together. Okay, so now we've got these disaccharides in because we digested them from starch, or that was in our food. It's the brush border enzymes that do the final digestion into these single sugars. So glucose, fructose, galactose. If we're digesting DNA or RNA, we get ribose. So these single sugars are now finally digested by the brush border enzymes. The combination of pancreatic enzymes and brush border enzymes. It's in trivial digestion, maybe in your saliva, but mostly it's in your small intestine. It's pancreatic enzymes, pancreatic amylase, and brush border enzymes. Okay. So to summarize then, the hydrolysis of polyglucose, starch and glycogen, may start with salivary amylase, but probably trivial because we don't get them out long enough. And as soon as we swallow it, the amylase is going to be denatured like any other protein in gastric acid. So it's gone. It's no longer functional as you swallow it. The pancreatic amylase that we secrete in the duodenum and that continues to work as it travels down the jejunum is hydrolyzing this down to maltose and maltriose and some glucose.
What is the jejunum? Oh yeah, yeah. It's breaking up the small intestine into 3. Right. Okay.
Brush border enzymes do the final breakdown of these ditrisaccharides into monosaccharides, including maltose, maltose, sucrose, lactose. And the brush border transporter proteins are then absorbing these monosaccharides, these single sugars, by active co-transport mechanisms.
So wait, let me read this. Pancreatic amylase and duodenum and jejunum hydrolyzes polysaccharides into maltose and maltriose. Brush border enzymes hydrolyze tridisaccharides into monosaccharides. And then brush border transporters absorb monosaccharides by active co-transport into hepatic portal. Hepa means liver, right? I'm pretty sure, but I don't think I have the root.
And from the hepatic portal vein, it travels to the liver, and now what happens depends on the insulin concentration. If insulin levels are high, then the liver takes up those sugars and stores them as glycogen. If insulin levels are low, it lets it continue on and travel into the bloodstream to raise your blood sugar back up.
Wait, the function of insulin is what? Okay.
Okay, so that's general carbohydrate digestion. So when you're talking about starch or glycogen, the digestion is fairly straightforward. These other weird polysaccharides, and it's a lot harder. For proteins, again, nothing happens in the mouth because there's no peptidases in saliva. Once it hits the stomach, they're unfolded because the acid pH is denatured. So that's physically opening them up without hydrolyzing them, without digesting them. And then pepsin can start the breakdown process.
Well, they don't really form HCl.
Into smaller pieces of chains of amino acids called polypeptides. Now the pancreatic juice includes both proteases and peptidases. So proteases again chewing on proteins, peptidases chewing on the smaller fragments of proteins, peptides. Plus brush order peptidases. Because unlike polysaccharides, which are all made of chains of sugars, so you leave the bond between two sugars, it's the same bond, proteins are made of dozens of different amino acids. And so the linkage between different amino acids is going to be different. So you need different enzymes to leave different combinations of amino acids. So you need more enzymes. So we have this combination there of proteases and peptidases that are breaking things down into smaller fragments. So smaller fragments are oligopeptides. So polypeptides are large fragments, oligopeptides are small fragments. Until you finally get into dipolypeptides, so 3 or 2 amino acids or just 3 amino acids. Okay, so we're whittling around these long chains of amino acids into smaller pieces. So from proteins, into polypeptides, oligopeptides, tri-amino acids, amino acids, dipeptides, tripeptides, and 3 amino acids. Now the brush order transporters are absorbing these ditripeptides and amino acids into the cells. So it's not just taking the free amino acids, it's also taking these smaller fragments. But the final digestion happens intracellularly. So it's actually reasonably new thinking that that's not like carbohydrates, where all the digestion happens in the lumen of the intestine. But the final digestion of proteins happens inside the cells. So intracellular digestion. But the final breakdown of these 2 or 3 amino acid pieces down into individual amino acids. Okay, so that's different. And then all these amino acids are then transported to the hepatocyte vein, and off the liver they go. And once again, it depends on insulin. If insulin levels are high, then it's okay for the liver to take up those amino acids and either use them to build proteins or to turn them into sugar and store them for later. Whereas if insulin is low, the amino acids travel into the bloodstream so other cells can use them. Okay, so that's protein digestion.
You would think that insulin wouldn't control protein or amino acid uptake by the liver. You would have. I would guess that it would be separate, but apparently it's not.
Now, lipid digestion is a little different. For a few reasons, one of which, of course, is that lipid is not soluble in water. So we need to play some games with it, be able to get to it, to be able to digest it. So, first, something happens in the saliva. It's a little bit like base of your saliva, but that's to keep it clean. It doesn't significantly break down the amount of lipid in your food. Nothing happens in the stomach. There's no like base in the stomach. Acid doesn't affect it. So nothing really happens to it until it gets to the small intestine. Now here's where the bile salts come in, those detergents, those cholesterol derivatives. start binding to this lipid and breaking it up into smaller and smaller droplets called micelles. Now, once it's exposed, now the enzymes from the pancreas can start chewing it up. So these lipases, so lipid digesting enzymes, right? We're all enzymes are proteins, but it's a protein that's digesting lipids, lipase. So pancreatic juice then introduced into the duodenum, continuing on into the jejunum, hydrolyzes these triglycerides. So fats and oils are what we call triglycerides. So it's not a chain of fatty acids. It's fatty acids that bind to a glyceride. So you have 3 of them on there. It's like a fat. That's a triglyceride. If you take one fatty acid off, you're left with a diglyceride. You take 2 of them off, you're monoglyceride. You take all 3 of them off, you're left with glycerol. Okay, so hydrolysis of fats. You're not breaking a polymer down into monomers, you're breaking the monomers off from the backbone. So these monoglycerides, so if you take 2 out of the 3 fatty acids off of glycerol, but one's still there. So monoglycerides and fatty acids, plus other fatty things in your diet, like cholesterol, all bind with these bile salts to form these micelles, these tiny little droplets. So we emulsify them into fat droplets, and now we're emulsifying them further into even tinier little droplets called micelles. Now there are other brush butter transporters that absorb these micelles in their whole. The whole micelle is absorbed into the cell. And again, the final digestion is happening intracellularly. But interestingly enough, they don't just digest lipids, they actually build lipids. So they resynthesize triglycerides. They put the fatty acids back on the glycerols in the way that they want to build them. Okay? So now you've rebuilt these lipids inside the cell. So this intracellular activity going on here was appreciated.
Chylomicron.
Now, how are you going to distribute this in circulation? Because it's not more soluble, right? These are fats. The answer is you hitchhike them onto a protein. Okay, so you have a carrier protein that's going to travel in the blood. You attach these fats onto the protein so that the protein can carry them in the blood. So that's how you transport fats in the blood. So you hitchhike them onto carrier proteins. Both triglycerides and cholesterol you absorb as well. And so the form of this, so a lipid on a protein is called a lipoprotein. Right? So lipid attached to a protein is a lipoprotein. Now the type of lipoprotein that your intestines makes is called a chylomicron. Okay, they're just coming next year. Chylomicrons are proteins carrying lipids and cholesterol from the gut to the rest of the body. Now there's a problem though here, so we had a problem that, okay, the triglycerides weren't going to travel in the blood by themselves, so now we're going to put them on a carrier protein. But now how do you get it in the blood? These chylomicrons are too big to pass across the walls of capillaries. But they will fit across the walls of the lymphatic vessels. That's what happens. The chylomicrons are absorbed in the lacteal. The lacteal doesn't go directly to the liver, it goes into the heart and into the general circulation. So the lipids that from your diet go directly into the systemic circulation. They don't go to the liver, doesn't get the first choice of those. Then as these chylomicrons are traveling down your blood vessels, tissues that need lipids, the cells lining the capillaries have enzymes on their surface that kind of harvest the lipids off chylomicrons as they go by. So how do we need some cholesterol here? How do we need some of that nitrate? They go directly into the systemic circulation. They don't go to the liver, doesn't get the first choice of those. Then as these chylomicrons are traveling down your blood vessels, tissues that need lipids, the cells lining the capillaries have enzymes on their surface.
The cells lining the capillaries. Oh.
That kind of harvest the lipids off chylomicrons as they go by. So how do we need some cholesterol over here? How do we need some of that triglyceride over here? And they're harvesting it off the carrier protein as the carrier protein goes by. So the chylomicron doesn't go into the cell, it stays in the bloodstream. The cells lining the capillaries are digesting those materials. They're not digesting, they're absorbing those materials off of the chylomicron as it goes by. So this is kind of a diagram of all of that. So here's again the lumen of your gut, where your triglycerides, your fats and oils in your diet, are being broken down by, so they're being emulsified by bile salts and hydrolyzed, digested by lipase into fatty acids and monoglycerides. The brush border transporters are absorbing these fatty acids and monoglycerides into the cells, where they're resynthesized back into lipids, along with other fat-soluble things like cholesterol. The triglycerides are then hitchhiked onto specific proteins to form the lipoproteins called chylomicrons. Those are exocytosed into the bacteria.
Oh shit.
Along with other fat-soluble things like cholesterol. The triglycerides are then hitchhiked onto specific proteins to form the lipoproteins called chylomicrons. Those are exocytosed into the bacterial hepatic vessel, and then back to the heart and circulation. Okay, so in general, this is how lipids travel in your bloodstream. So again, here's sort of just some general lipoproteins found in your blood plasma. So here's the chylomicrons that come from the intestine, and their destination is organs all over the body, traveling triglycerides and other lipids from your diet to your organs. Now, if you've got excess, the liver does want to hold on to those. So there are some of these proteins then that will take excess liver. So if between meals you need more fatty acids in your bloodstream, your liver also has lipoproteins that it makes, called low-density lipoproteins and very low-density lipoproteins. Because you know oil floats on top of water, so the more fat you've got, the more fat-to-protein ratio you've got, the lower density it is. And so these travel then from the liver, so this is how the liver sends fats to the different organs. So the triglycerides or the low-density ones are specifically carried to the liver enzymes. Okay, so in general, this is how lipids travel in your bloodstream. So here's sort of just some general lipoproteins found in your blood plasma. So here's the chylomicrons that come from the intestine, and their destination is organs all over the body, traveling triglycerides and other lipids from your diet to your organs. Now, if you've got excess, the liver does want to hold on to those. So there are some of these proteins then that will take excess liver. So between meals you need more fatty acids in your bloodstream. Your liver also has the lipoproteins that it makes, called low-density lipoproteins and very low-density lipoproteins. Because you know oil floats on top of water, so the more fat you've got, the more fat-to-protein ratio you've got, the lower density it is. And so these travel then from the liver, so this is how the liver sends fats to the different organs. So either triglycerides or the low-density ones are specifically carrying cholesterol. On the other hand, if the cells already have too much cholesterol, the cells can produce a protein that will carry it from back to the liver. And so those have less lipid on them, so these are high-density lipoproteins. Those are taking cholesterol from the tissues back to the liver. Okay, so we talk about good cholesterol, bad cholesterol. There's only one kind of cholesterol. It's which bus is it on? Is it going into the tissues or out of the tissues? Because if you're especially your blood vessels, if they get too much cholesterol, they get stiff, and they're more likely to form clots and therefore cut off your circulation, which is heart attacks, strokes, things like that, related to too much bad cholesterol. Meaning cholesterol only has LDLs and VLDLs that's being put into your cells, and then not enough of HDLs is being taken
Oh wow, that's what LDL is? That's crazy.
down your cells. That's why it's the ratio of LDLs to HDLs that's more important than natural cholesterol. Okay, so LDLs are lousy, HDLs are healthy. So HDLs tells you that you're removing cholesterol from your tissues. LDLs means you're dumping cholesterol into your tissues. Right, all the same cholesterol, which bus are they on? So here's just a diagram of the sort of ratio of, again, these biodetergents, lipids, triglycerides, cholesterol molecules, and these proteins that are allowing them to travel in the bloodstream. So here's a chylomicron that we made by the intestine, some LDLs and VLDLs that we made by the liver to distribute more liver. Cholesterol, more lipids to the tissues, and then these HDLs that have a much higher protein to lipid ratio, so they're higher density, that are removing cholesterol from the tissues and getting them back to the liver. Okay, so that's the small intestine. So small intestine, particularly the jejunum, is where most of our true digestion and absorption of nutrients happen. And for proteins and carbohydrates, they're sent to the liver, and then if insulin levels are high, they'll travel on to the rest of the body. Whereas for fats, it goes in lacteal into the general circulation. Now in the ileum, as well as now in the large intestine, the colon, the main function is to absorb water and salts in the diet. And since there's a lot more water and salt, you need a lot more gut absorbing all that so you don't dehydrate yourself as you're trying to digest your food. So the colon is where it's regulated. So in the ileum, it's pretty much automatic. You're going to get back a certain most of the water there. And now in the colon, you're more regulating, okay, how much water do we really need to get back? So again, there is a valve to prevent material from the large intestine, from the colon, to go back into the small intestine. And mostly now we're doing our final absorption of water and salt. So we're compacting and storing and excreting the feces. Now because your anus is up in the outside world, it's not too surprising it's colonized by microorganisms. And so it's microflora of bacteria and protists and even some microbezoans are living in there and feeding off of whatever you didn't digest. So whatever you couldn't digest, that's now feeding all these microorganisms. Now a lot of these microorganisms are beneficial, particularly if you have a healthy population there. Number one, they help repress other bacteria that might not be so healthy from coming in. But as a side product of their metabolism, they make actually some vitamins that we can use. And even some of them, even though we may not absorb them in our bloodstream, they're healthy for the cells of the large intestine. And so when you're pooping out your feces, about a third of the mass of the feces you poop out is actually bacteria from your colon. So it's not your undigested food, it's things that are living in your gut. Okay? But the brown color of the feces is from those bile pigments, those breakdown products of hemoglobin. Okay? That's also what makes your urine yellow, these bile pigments. Okay, so some of it, that's what gets back into your circulation. So again, here's the ileum of the small intestine that plugs into the cecum, the pouch down here. And then at the end of the cecum is the appendix, which is more of an immune organ. It's kind of sensing what kind of organisms you're being exposed to here. Okay, so some people get the cecum and the appendix confused. It's like the appendix is a little tiny thing, the cecum is like a pouch. And again, herbivores, that cecum is large, and carnivores is short, and omnivores is kind of in between. And then from there goes ascending, transverse, descending, sigmoidal, curved rectum, anus. Okay, and when we talk about kidney function, the regulation of water and salt reabsorption of the kidney is regulated by the same hormones that are regulating the large intestine. So both of them are involved with reclaiming water and salts and maintaining osmolar altitude. Next week's subject. So our overview then for carbohydrate digestion, a little bit starts in the mouth, mostly it's in the small intestine by pancreatic amylases, and then the brush border enzymes that do final digestion. Proteins, nothing's happening in saliva. In the stomach, though, you get denaturation and some digestion. Most of the digestion happening from a whole bunch of different proteases, peptidases, in the pancreas. Then the final digestion happening intracellularly in the lining of the cells, in the epithelial cells lining the intestine. Nucleic acids we didn't really talk about much because proportionally it is a macromolecule, but it's not that big of a percentage of your diet. But we do certainly have pancreatic nucleases that break DNA and RNA into its monomer nucleotides, and then enzymes that break the nucleotides down into sugars and nitrogenous bases and phosphates, so it's components to it, which they're transporters that can absorb those. And then fats, again, the bile salts are emulsifying them into droplets. The lipase then breaking them down into glycerol, fatty acids, and glycerides. And then intracellularly being resynthesized back into triglycerides and hitchhiked onto lipoproteins. Okay, so that's pretty much the pattern in our bodies. In other mammals, there are similarities, but with differences depending on what the types of things that they're eating. And so, as we've said several times, that plant material is much harder to digest, not because starch is easy to digest, and of course, sucrose is easy to digest, but it's the other complex polysaccharides and odd sugars that plants make that are just they're not easy to break down. And so in many cases we can't break them down ourselves. We need help from microorganisms, protists and bacteria that have enzymes that we don't. And so again, in general, in herbivores we see a much more longer and more complicated gut compared to a carnivore. So carnivore, short intestine, small cecum, pretty simple cecum, and large intestine, so fairly overall short gut, short cecum. Now, among the herbivores, there's one group of mammals that's really good at digesting terrestrial plant material, and that's a particular order of mammals called the ruminants. And the ruminants, their stomach is divided into these 4 chambers, and that allows them to host a whole bunch of microorganisms right at the beginning. So they're able to ferment that food, and then pretty much they're farming the microorganisms. They're actually living more off the microorganisms than they are off.
Rumen, reticulum. So culum we know is a diminutive suffix. We learned that yesterday, but omasum, abomasum.
Then they are directly off of the grasses and leaves, their grazers and browsers, that they're feeding on. And then a really long intestine to digest all that material, a really big cecum with more fermentation going on, and then a really long intestine, large intestine, because it takes a whole lot of water to do all this digestion, and so we need to reclaim a whole lot of that water before we finally excrete it out the other end. So the ruminants are really good at digesting terrestrial plant material because they've got this very specialized gut. So if we look at it a little closer here in our cow, so again, the masticated vegetation, so they're doing oral mastication, swallow into this first big chamber here called the rumen. And the rumen is where a lot of this fermentation is going on, so these microorganisms breaking down the cellulose. And every once in a while she regurgitates and chews it some more, sends it back down, helping out those bacteria. And then these other chambers of the stomach. The reticulum then is kind of filtering it, so all the grass, bigger chunks are staying in the rumen, and the smaller pieces are then filtering through into the omasum and abomasum, and then finally into the small intestine. Okay, so most of what it's actually swallowing is this huge population of microorganisms living off of this masticated grass. So the cow is a farmer. She's farming these microorganisms. Now, other herbivores don't have it. So this rumen is a very specialized structure. And there's certainly more animals out there that are feeding on plant material than there is than just the ruminants. And so they are also relying on these microorganisms. But those are located where most microorganisms are in animals. In the cecum and large intestine. So here again is an insectivore. So this shrew here is a strict carnivore. It feeds pretty much exclusively on insects and worms. So fairly short gut with not a whole lot of elaborate specializations. Whereas this herbivore, this non-ruminant herbivore, now has this much longer gut. And so most of their fermentation is happening in the cecum, kind of like it was in the insects. But remember, in the insects, the cecum was at the beginning of the midgut. This is after the midgut. So the problem here is that you're digesting all this food, but you can only absorb it in the small intestine. So how does that do you any good? So the cellular protists and bacteria are in the cecum, but it's after the junction with the small intestine. And the answer is that even though this is carbohydrate, this plant material, these cell walls are carbohydrate, so the small intestine is doing regular digestion of other things, right? Proteins and simple carbohydrates like starches, fats. Those are all digested normally in the small intestine like they are in any other mammal. But it's these complex carbohydrates that animals can't digest that are now being processed by these microorganisms in the cecum and large intestine. But it turns out that the particular metabolic pathways of these microorganisms takes these chains of sugars, these complex carbohydrates, and metabolize them into volatile free fatty acids. And volatile free fatty acids is one of the few organic molecules that can be absorbed by the large intestine. Now in us, our guts do the same thing, but they don't really get into our circulation. But they are good for the lining to our gut. So that's one of the reasons why soluble fiber is so important in our health, is because it keeps our gut happy, keeps our large intestine cells. Our large intestines are much healthier, less likely to get various diseases, including even colon cancer. Less likely if they get a healthy dose of this fiber, that these microorganisms are then able to nourish those cells. So it's good for the nourishment of those cells. In the herbivores, they're producing so much of it, they're actually getting into their circulation, getting nutrition from these. Okay, so we get this large cecum. Now, one of the other methods So, okay, we're saying we're digesting all this material, and some of it's, a lot of it's being produced in these fatty acids that can be absorbed. But still, a lot of it's just being processed into other carbohydrates that maybe you can now digest them, but they're the wrong end of your small intestine. So how do you get them back up in your small intestine? I'm going to eat your poop. So it's called coprophagy. So coprophagy.
Bro, you guys know what I'm gonna say. Y'all do this. I know you do. Don't even try to freaking lie. I know y'all do this.
He's poop eating. So here's this cute little bunny, and then here's the stomach. Here's chewed up grass that's been consumed, and then here's a whole bunch of re-ingested feces. Right, so this cute little bunny. And these are poop eaters. Right, they send it through a second time. Okay, the microorganisms had a crack at it. They broke it down to stuff that now I can digest. Okay. So again, even so, carbohydrate is just hard to digest. Even in a ruminant, like this wildebeest here, that 45% of the cellulose is broken down, which means 55% of it isn't. So quite a bit of it goes all the way through without being digested, even with a rumen. So in something a non-ruminant like a bunny, it's like there's a whole lot of energy and resources there that didn't get absorbed.
What are y'all up to this weekend? Coprophagy? Probably. It's probably what you're up to.
Okay, let's do a little review of a non-ruminant herbivore, the horse, and the digestive processes that we find there.
We start with the horse eating a meal that contains protein, fat, and carbohydrates, including starch and fiber. Chewing is the first stage of digestion, mechanically grinding the food, making it more available for digestion as it progresses through the tract. In the horse, chewing also stimulates the production of saliva, which lubricates the feed as it slides down the esophagus. Notice that the horse does not chew up and down like humans, but chews in a grinding motion. Dental care is vital in horses to help maintain this proper chewing motion. The horse swallows the feed, and the feed bolus flows down the esophagus and into the stomach. Due to the sharp angle where the stomach and esophagus meet, and the strength of the esophageal muscles, horses are...
Did I have bolus in here? I think it's just like a piece of food.
...are not capable of vomiting. In the stomach, feed is broken down into starch, the yellow beads, fiber, the green beads, protein, the red blocks, and fat, the white globules. Notice the digestive enzymes released from the pancreas as nutrients enter the small intestine. The horse's small intestine is 70 feet long, and feed flows through it for roughly 45 minutes. This is the primary site of enzymatic digestion. Starch, protein, and fats are all digested here, as well as the absorption of amino acids. Normal digestion and absorption, carbohydrates in the upper gut. First, let's follow starch, the yellow strings of beads, and fiber, the green strings of beads. Alpha amylase enzymes from the pancreas enter the small intestine and break starch molecules into disaccharides, which are two glucose molecules bound together. Notice that the fibers flow on intact through the small intestine. There are no enzymes produced in the mammalian digestive system capable of breaking the bonds between the sugar molecules in fibers. As the disaccharides continue flowing through the small intestine, they bind to amylase enzymes in the lining of the intestine. These enzymes catalyze the reactions to break the disaccharides into their
So those are the brush border, right?
simple glucose components. Glucose is then absorbed through the intestinal wall into the vast blood capillary network that surrounds the gastrointestinal tract. In the blood vessels, glucose molecules, the yellow spheres, are transported to body tissues that will utilize them immediately for energy or to store for later use. Insulin is released from the pancreas in response to rising glucose, and it stimulates the movement of glucose out of the bloodstream into tissues. Insulin binds to receptors on the plasma membrane and allows glucose to move from the blood into tissues, where it will immediately for energy or to store for later use. Insulin is released from the pancreas in response to rising glucose and stimulates the movement of glucose out of the bloodstream into tissues. Insulin binds to receptors on the plasma membrane and allows glucose to move from the blood into tissues where it will be used for energy or other metabolic purposes, such as being stored as
Not again, bro. They just move back and forth, dude.
Glycogen, or used in the synthesis and storage of fat. Normal digestion and absorption, fats in the upper gut. Digestion and absorption of fats differs from other nutrients because fats are not water-soluble. Unlike most species, horses do not have a gallbladder, but they're very efficient at digesting fat. Bile produced in the liver is continuously secreted into the horse's small intestine. Bile salts surround and emulsify the fat, making it available for enzymatic digestion by lipase from the pancreas. The resulting lipid products combine with bile acids and other molecules to form micelles, which then diffuse into the cells of the intestinal lining. The absorbed lipids are then repackaged with cholesterol and proteins into kilomicrons. Kilomicrons are too large to be absorbed into blood capillaries, so instead, they're expelled from cells and are taken up by the intestinal lymphatic vessels. They travel through the lymphatic system and finally reach the bloodstream via the thoracic duct, where the lipids can be used as fuel, incorporated into other compounds, or can be stored as fat. Normal digestion and absorption of proteins in the upper gut. Enzymatic digestion of protein begins in the stomach with the enzyme pepsin, which breaks protein into shorter chain peptides. In the small intestine, pepsin, trypsin, and several other enzymes from the pancreas and stomach further break down peptides into the specific amino acid building blocks, and these amino acids are absorbed through the intestinal lining into the blood capillaries. Amino acids are carried through the blood to the tissues, where they are used to build proteins for many functions, including building and repairing tissues, components of enzymes, antibodies, hormones, and blood. Normal digestion and absorption, fiber digestion in the hindgut. The hindgut of the horse is about 25 feet long, but represents about 60% of the volume of the horse's digestive tract. The primary digestive role of the equine hindgut, which includes the cecum and large intestine, is the digestion and absorption of fiber. The cecum is located at the junction of the small and large intestines and contains billions of microbes, such as bacteria and protozoa. These microbes produce the enzymes that break the linkages between glucose molecules and fibers. The end products of microbial digestion or fermentation in the hindgut are volatile fatty acids. These volatile fatty acids, or VFAs, are absorbed through the lining of the large intestine and are an important source of calories to meet maintenance energy requirements of the horse. Excess starches and sugars that are not digested in the small intestine are also fermented by microbes in the hindgut. But again, the results are volatile fatty acids, not glucose molecules. The only way to get glucose from fermentation in the hindgut is for 1-VFA, propionic acid, to be repackaged into glucose in the liver. This process is called gluconeogenesis. Proteins are also digested by microbes in the hindgut, but microbial digestion does not result in absorption of intact amino acids. Microbial protein digestion produces nitrogen and carbon skeletons that may be used by the body or excreted as ammonia. For horses to meet requirements for essential amino acids, they must be supplied by the diet in proteins digested and absorbed in the upper gut. Normal digestion and absorption, completion of digestion. In the large intestine, as muscular contractions move the digestion through, digestion and absorption of nutrients continues, as well as the reabsorption of much of the water from the food mass. The microbes also produce some nutrients, such as B vitamins, some of which may be absorbed from the large intestine. The muscular contractions and reabsorption of water in the large intestine and colon results in somewhat dry, rounded fecal balls, which you then clean out of your horse's stall. It may be roughly 3 days before everything you feed makes the trip through the digestive system and onto the floor of your stall.
Okay, so that's the general mammalian pattern. We see similar things in most vertebrates. But there are some issues that some vertebrates, non-mammalian vertebrates, do have to deal with. So we have some similar things, for example, in herbivorous reptiles.
Bro, we've got to get the phylogeny down. Koano? I don't know if I have Koano. Porifera. Metazoa. But I think the protostomes, it's not like a good name, right? Like I think even if you just look up, let's close all this stuff. Once thought to be characterized by the formations of the mouth before the anus, this nature has since been discovered to be extremely variable. That's actually kind of wild. The radial symmetry as adults thing is pretty crazy. Okay, so what are we trying to figure out though?
Plant material. That is primarily the cellulitic, in other words, can break down cellulose, protists and bacteria in an enlarged colon that does most of their fermentation. And so they've got a huge stomach because you have to eat a tremendous amount of this food. So if you consume a large amount, then digest it slower, and then a huge colon where they're fermenting all this material. And again, it's not breaking these carbohydrates down into sugars because they can't absorb these from the large intestine. They have to be metabolized by these microorganisms into these volatile free fatty acids, and those can be absorbed by the large intestine. But the caveat to this is that those microorganisms require a warm temperature to do it efficiently. So in homeotherms like mammals, that's
Bro, and like all of a sudden, like all of them leave. It's crazy, man. They move so quickly. I don't know what caused them to move. Oh, now they're coming back. Crows are so weird, dude.
not a problem because our bodies are always warm. But if you're in a coelotherm like a reptile, then it's going to depend on the environment. So pretty much you only find herbivorous reptiles in the tropics. And so like this iguana here or the green sea turtles, those are herbivores, but they're primarily restricted to the tropics. You start getting away.
Therefore, ectothermic herbivore reptiles are limited to tropics and deserts.
From the equator and the temperate regions, and it's just too cold. Primarily the reptiles in those areas are going to be carnivores. Right? So you can start moving up into these latitudes up here, and the only sea turtles you see are usually leatherback turtles, which are feeding more on jellyfish and not on algae. The same thing with the reptiles we get up here. We're getting more carnivorous reptiles than carnivorous lizards and snakes. Of course, all snakes are carnivores. Then you are herbivores. So limitation then of poikilothermic herbivores. And then some animals do have the ability to make their gut more efficient when they need to be. So for example, if you're in a temperate environment, in the wintertime there's not a whole lot of food. And so there's some birds, like these wrens, that their gut can actually get 20% longer in the winter. So whatever food they do find, they're more able to extract every last bit of nutrition they can out of it. Or mammals when they're lactating, right? When you're eating for two, when you're pregnant and when you're lactating, mammals also, sometimes many mammals, their gut will actually get larger, females, when they're lactating, as well as just eating more. So all of this relates back to when we talk about thermoregulation, that we have energy budgets, that we only can consume so much energy, and we have things we need to spend that energy on. So we need to spend it on growth, on maintenance, on just running all those pumps that are keeping our cells at the right voltage, and reproduction, and for homeotherms that keep our bodies warm. So
Right.
that energy has to go somewhere. So the amount of food we eat is going to determine then how much we have to spend. And so particularly in herbivores, that it takes a lot of food to be able to get enough energy out of it. So you look at this caterpillar here, and for every 200 joules, so joules is a measure of energy, kind of like calories, for every 200 joules, only half of that is actually being digested. Half of it's just being excreted right out the other end as feces. And then out of the 100 joules that it did get at that 200, 2/3 of it's just going to keep itself alive. There's only 1/3 of it's available for growth. So once this caterpillar, which is a juvenile, becomes reproductively mature, it can't afford to grow anymore. So that's why most insects, once they become mature, stop growing. You just can't get enough food out of your environment to support both growth and reproduction. So once we've absorbed this nutrients from our food, we can circulate it. So some of it travels in our blood. We can store it. So again, in vertebrates, we store glycogen. That's the form of carbohydrate that we store, our polysaccharide, in our muscles and our liver. Although our liver is the only one that can turn it back into blood sugar. So when the muscles take blood sugar and turn it into glycogen, that's for their use. They can't put it back in the blood. Or some tissues can take sugars and turn them into fat and store them as fat. And again, fat has a much higher calorie density to it, so that's why animals prefer to store their energy as fat, because you get more energy stored per volume of tissue. And then you gradually use that for just keeping yourself alive, your overall metabolic processes, running all those ion gates, breathing, respiring, doing all those things you need to do. And if you're a homeotherm, keeping yourself warm, then to grow. And again, as we said, most insects, once they turn reproductively mature, can't grow anymore. And reproduction. And so again, it's organic food molecules of whatever you've ingested. What you can't digest is going to be lost as feces, so passed on to the detritivores and decomposers. What you can digest then into its monomers, you can either use as fuel to make ATP, or you can use those as building blocks to build new macromolecules. And then the ATP we need to power that synthesis, or to power all the other things we need to be alive. Eventually breaking, or doing all those energy transformations, is going to transform that energy that's tied up in the chemical bonds of our food into heat lost to the universe. And so we constantly need to replace the energy that's being emitted away into new energy coming in. And so different ways that we store, different ways that we circulate, different ways that we use these macronutrients. So energy reserves can be stored as fats, triglycerides, as glycogen, polysaccharides, or as proteins. We don't deliberately store proteins for energy, but if we need to, we can break down our proteins for energy. So this could either be from the gut or from where we're storing it. We then break our fats down into fatty acids and glycerol, and if we need to, the liver can produce something called ketone bodies. So those are the circulating forms of lipids. Polysaccharides can be broken down into blood sugar, glucose, or if they've been used for anaerobic respiration as lactic acid, and proteins into amino acids. So these are the circulating energy substrates.
Oh, this is tissue, this is a blood vessel, and then this is intracellular? I see.
Then cells take up preferentially different of these. Different tissues have different preferences, and we can use the sugars to go through our metabolic pathways to produce ATP. We can use fatty acids, they plug in here, amino acids plug in here.
Krebs is oxidative or no? What does it say? Okay. Whoa.
Here and here. Ultimately, whatever these organic molecules are, we're producing carbon dioxide and water and ATP and heat. Okay, so this is how we store them, this is how we circulate them, this is how we use them. And we'll go into a lot more detail on this in 6b. So that's staying adequately nourished. Malnutrition is an imbalance, either too little or too much of either total calories or specific types of nutrients that you use, macronutrients or micronutrients. Either too much or too little. And it's kind of odd that our society seems to oscillate between undernutrition and overnutrition and rarely manage to balance itself at just well nourished. Okay, so that's our brief little trek through the process of animal feeding and nutrition. And again, we'll talk about the using of those resources applied to other systems and other balances as we continue our discussion and continue.
All right. Let me do this. Okay. Hydra. Yeah, Hydra is a Nidarian, I think, right? Yeah, Nidaria, which is what? Radialis or whatever. Radiata. It's not even in here. Okay, so it's definitely this though. What? Wait. Radiata. I don't think this is the right slide. I think it's circulation. It's not in circulation. Actually, they're probably filter feeders, right? Isn't that what it said? Oh no, it doesn't even say that as an answer. Oh, that's a good question, actually. Let's see. Was it in develop? Well, so this is before bilateria. So only the bilateria is the only ones with mesoderm. Seal information. It might not even be in there. It might be in the lab lecture. Oh wait, we didn't get them all. Wait, oh no, wait, hold on. So animals are defined by blastula. So all of these guys, the sponges and the cnidarians, do blastulation. They just don't do. Which stage does gastrulation then? The eumetazoa. So the kind of idea is like once you have mesoderm, you get an explosion of differentiation or something like that. Okay, so we're talking about the diploblasts. Yeah, which the chat is calling radiata, but diploblasts. Okay, variation in gastrulation. So okay, so the sponges. Okay, so all of these guys have gastrulation. So it's really only the sponges that don't have form a gastrula. I thought for sure that these guys had a gastrovascular cavity. Okay, so here are the variations. So we have the blind gut, the protostomes, and the deuterostomes. Yeah, bro, isn't this a cnidarian? Does that say cnidarian? Oh no, this one's cnidarian. So we have metazoa, true metazoans. True metazoans have gastrulation. So they have some sort of a gut. And even in this other one, I hate that the top here isn't the same species as the top here, but that's fine. Wait, sponges are diploblastic, radiosymmetry, no coelom. Okay, let's use this one as a guide. Porifera, right? Porifera. They still have blastula stage, but they have no gastrulation. Okay. Okay, so that's what they call the spongiceal, but it's not really like. Okay, and then there's diversity in that. That's fine. Okay, here we go. Cnidaria. Yeah, what the hell? Nidaria, gastrovascular activity. I guess digestion must not happen in there. Digestion must be intracellular. Because Hydra's a polyp, right? Let me get some food, I'm hungry. They're solitary carnivorous jellyfish-like animals. Okay, the word gastrovascular is not in here. Where is feeding? Okay, so the mouth of the hydra is surrounded by 4 to 8 tentacles. When feeding, hydra extended at a maximum length of solution. Tentacles are extensible. Search for prey. Neurotoxins. Whoa, that's kind of crazy. So the question is more specific. Where does digestion occur? Let's ask chat. I probably get this wrong too, because I feel like this is a trick question. Okay, Hydra has a gastrovascular cavity. Do all... Let me write out... Well, I don't have my pen. Do all... What? Wait. Oh, this is Hydra. We need to go to the... Whatever. It doesn't matter. So platyhelminthes, some non-radiate bilaterian animals also have a gastrovascular cavity, even though they're triploblastic and bilaterally symmetrical. Okay, let's try. So then, we should find where does he talk about feeding then? Is it in the feeding slides? Oh, here we go. Digestion. Let me pull up the lecture then. It's feeding, not... Oh, wait. Is there separate slides for that? No, I think it's all one. I think there's just 2 lectures. Oh wait, wrong one.
Stand out, measure like white. And so when the British came in and kind of kicked the Dutch around, they thought they were saying white rhino. And so that's what that actually because rhino doesn't see those enzymes. Now the membrane of the lice digests other proteins. Lipase digests proteins like lipids. Lipase digests lipids. Things like ASE, suffix. And those we're talking about enzyme. The first word says what is digest enzyme. Specifically not digest enzyme. No, all the way. So it's alcohol. So you look for this means. Right now, it says membrane. And there was in here these besides exocytosis. I love that. So. Okay. So how do you digest your food? It can be from it. The size of never actually seen food. I'll never actually seen digestive enzymes. All of that's coming inside special parts. And of course, this is how their whole system works. Here's a seen that silly referred to in class. That has essentially got single words. How does that work? Well, so it's that cell which uses the little down, but also after eating food crops. This will move the surface of the cell.
Well, that's Hydra.
This is the groove, it's the groove. So the cell creates currents, little particles will get pure bacteria, or just like this groove. And after the groove is where endocytosis or phagocytosis. Then brings the food to the food cycle. You then have all the cells that infuse with it.
So then on the next slide, he should have the answer.
And then finally, whatever you then make with that. Special memory that does all these functions over and over again. So okay, we've done that out. Let's go get some more stuff over here. Now, there are also, generally we talk about animals having extracellular digestion, but that's not always true. There are some animals, and most animals do a little bit of both, but there are some animals that primarily do intracellular digestion, and there's good reasons for that. So the sponges, we talked before about sponges, how they create this current, all this gut here is surrounded by all those flagellated cells that create that current that pulls water in through these pores, catches food, and then the food goes out this big pore over here. So they tried to do extracellular digestion and did dump their enzymes out here, all that stuff just gets swept away. So that doesn't really work for the sponge. So the sponge then has these collar cells, these choanocytes, that have these sticky collars around the flagella. So the water that comes in through the pores is filtered through these little finger-like, we call them microvilli, which are covered with mucus. That mucus then traps the food. They then phagocytose those captured food particles, digest them, consume them, or they can actually pass those vacuoles, those food vacuoles onto other cells that can then distribute the food to the rest of the body. Okay, so intracellular digestion in an animal. And then some of our cnidarians as well, the hydra here, again, because it's got this gastrovascular cavity, it's doing both circulation and digestion, a lot of the digestion will be, so some of it will happen in the gut, so some of it's extracellular, but then a lot of the big pieces are taken up into the lining of the gut and digested intracellular.
All right. It's good enough for me. Ingestion. Oh, this is interesting. It's probably this one. It's got to be that one. It could also be this one, right? No. It's this one. Fat digestion yields fatty acids and glycerol, whereas protein digestion yields amino acids. Which of the following statements is true regarding this process? I actually don't know if that's true. This is definitely not true. I think this might just be proteins. And I don't know about that one. I'm gonna check this one. Okay.
The body obtains fuel, raw materials, and essential nutrients from food. A hamburger contains minerals, vitamins, water, and several kinds of large molecules: carbohydrates, proteins, nucleic acids, and fats. Large molecules must be digested, broken down to their smaller, simpler building blocks before they can be absorbed into the blood. Let's review digestion and absorption in detail. When large molecules are digested, the smaller molecules produced are monosaccharides, amino acids, nucleotides, and glycerol and fatty acids. Take a hamburger. A hamburger contains a mixture of carbohydrates, polysaccharides, disaccharides, and monosaccharides. Starch molecules in the hamburger bun are polysaccharides. Sucrose or table sugar in the ketchup is a disaccharide. The ketchup might also contain fructose, a monosaccharide. Among these three, fructose is the only nutrient that does not need to be digested further in order to be absorbed. Monosaccharides like fructose are ready to be absorbed, but the disaccharides and polysaccharides need to be broken down to monosaccharides by digestive enzymes. In the mouth, salivary amylase, an enzyme secreted by salivary glands, begins the digestion of starch molecules.
Actually, probably good to do salivary. Salt maybe come from salt. I actually have no idea.
Breaks starch all the way down to disaccharide units. Pancreatic amylase is contained in pancreatic juice, secreted by the pancreas when food is present in the small intestine. Maltose is the disaccharide produced when pancreatic amylase digests starch. Each maltose molecule consists of 2 glucose molecules. Disaccharidases, secreted by the walls of the duodenum, the first part of the small intestine, break down maltose, sucrose, and lactose from milk. The resulting monosaccharides are absorbed through the walls of the small intestine into the blood. Protein molecules must be broken down to individual amino acids in order to be absorbed. The digestion of proteins begins in the stomach with the secretion of gastric juice. Pepsin is the enzyme in gastric juice that begins the process of protein digestion. Gastric juice contains mainly water, hydrochloric acid, and the enzyme pepsin. Pepsin is secreted by the walls of the stomach in inactive form and activated when it comes into contact with the acid. Pepsin partially digests protein, producing short polypeptides. Protein digestion is completed in the small intestine. The pancreas is the organ that secretes most of the enzymes that continue protein digestion. Trypsin, chymotrypsin, carboxypeptidase, and aminopeptidase are the pancreatic enzymes that digest proteins. Trypsin and chymotrypsin break peptide bonds adjacent to certain amino acids. Carboxypeptidase and aminopeptidase break off one amino acid at a time, starting at the ends of a polypeptide. Dipeptidases
No, that's
attach
kind
to
of
the
random.
intestinal lining.
Is that really what happens?
Eventually, polypeptides are completely broken down into individual amino acids, which are transported through the walls of the small intestine into the blood. Food contains small amounts of nucleic acids, DNA and RNA, which have to be digested to be absorbed and used by the body. Nucleic acid digestion is carried out in the small intestine and is initiated by pancreatic enzymes. Enzymes called nucleases from the pancreas break nucleic acids down to their nucleotide subunits. Other enzymes attached to the intestinal wall further break down the nucleotides, and the parts are absorbed into the blood. Digestion of lipids, like that of other nutrients, mainly takes place in the small intestine. However, digestion of lipids, mostly fats or triglycerides, presents a problem. Because they are insoluble in water, fats tend to form large droplets that are hard to break up. The liver produces a fluid containing an emulsifier that disperses lipid droplets. Bile, produced by the liver and stored in the gallbladder, contains bile salts that coat fat droplets and keep them from coalescing. Smaller droplets present a greater surface area of fat for enzymes to attack. Lipase from the pancreas breaks fat molecules into glycerol and fatty acids. These pass into epithelial cells in the wall of the small intestine and are recombined to form fat. The fats
I wonder
combine
why they do that. Like, why do we recombine it?
cholesterol and coated with protein to form small
It's kind of
globules.
weird.
These enter lymph vessels, which eventually carry them to the bloodstream.
Okay. It might be maltose. But sucrose is a disaccharide, I'm pretty sure, right? It's either this or this. Is the video still open?
Carboxypeptidase and at a time be absorbed. The digestion of proteins begins in the stomach with the secretion of gastric juice. Pepsin is the enzyme in gastric juice that begins the process of protein digestion. Gastric juice contains mainly water, hydrochloric acid, and the
Bile, oh, that one's easy. They should have given me more bile ones. To emulsify fat in the small intestine, yeah.
Digestion takes place in the
10
alimentary
minutes?
canal,
Come on,
also
bro.
called the digestive tract or gastrointestinal tract. Basically, this structure is a tube that starts at the mouth and ends at the anus. Several vital organs and glands are connected to the alimentary canal and contribute digestive enzymes and other products to specific portions of the canal. Food is processed mechanically and or chemically, depending on where it is in the alimentary canal. Food is chewed or processed mechanically by the teeth. At the same time, it's broken down by enzymes or processed chemically by saliva from the salivary glands. The esophagus transports food to the stomach, where mechanical and chemical processing continue. Food then enters the small intestine, where chemical processing is completed and the absorption of nutrients begins. The liver secretes substances required for the digestion of fats, and the gallbladder stores these substances before emptying them into the small intestine. The pancreas secretes enzymes and other substances into the small intestine to carry out chemical digestion. Finally, what's left of the food is. The liver secretes substances required for the digestion of fats, and the gallbladder stores these substances before emptying them into the small
So
intestine.
the liver really is only for the
pancreas
bile
secretes
in
enzymes
terms
and
of
other substances
digestion.
into the small intestine to carry out chemical digestion.
But the pancreas has the
Finally,
amylase,
what's left of the food
protease, and
makes its way through the
whatever.
large intestine, where water is absorbed and feces are formed. Animals don't acquire nutrients as individual molecules, but instead must digest their food internally. They must extract nutrients from the food and eliminate wastes. Let's follow a meal as it travels through the alimentary canal. For simplicity, we'll divide our meal into its main macromolecules: carbohydrates, proteins, and fats. First, we'll look at the digestion of carbohydrates, which begins in the mouth. Both mechanical and chemical processing occur there. Chewing reduces the size of food particles, and the salivary glands secrete the enzyme alpha amylase. This enzyme cleaves the bonds in polysaccharides such as starch and glycogen, beginning the hydrolysis of these polymers to glucose very quickly. The salivary glands also release water and glycoproteins called mucins. Together, water and mucins form mucus, which makes the food slippery enough to be swallowed. Once food is swallowed, a wave of muscular contractions called peristalsis propels the food particles through the esophagus to the stomach. When food enters the stomach, muscular contractions churn it, mixing the stomach contents and reducing them to a uniform consistency and osmolarity. Most of the chemical processing that occurs in the stomach is the digestion of proteins, not carbohydrates. Peristalsis in the stomach walls moves small amounts of the partly digested material into the small intestine. There, secretions from the pancreas, including a form of amylase, are added to the mix. Pancreatic amylase continues the digestion of carbohydrates that began in the mouth. The cells lining the small intestine absorb the sugars that are broken down by pancreatic amylase. The epithelium of this organ is folded and covered with finger-like projections called villi. The cells that line the surface of villi have projections of their own called microvilli. The folds, villi, and microvilli of the small intestine greatly increase the surface area available for absorption. Nutrient absorption depends on the presence of an electrochemical gradient that favors the influx of sodium ions into the epithelium of the small intestine.
Favors the influx of what?
Nutrient absorption depends on the presence of an electrochemical gradient that favors the influx of sodium ions into the epithelium of the small intestine. This gradient is established by the active transport of intracellular sodium ions out of the epithelial cells, with ATP as the energy source. The movement of sodium ions out of the epithelial cells results in a low sodium ion concentration inside the cells. With this electrochemical gradient established, sodium-glucose co-transporters enable nutrients such as glucose to exit the lumen of the small intestine against their concentration gradient and enter the epithelial cells.
No.
Water follows passively by osmosis. The absorbed glucose exits the epithelial cells and enters the bloodstream, which carries it throughout the body. By the time the processed material reaches the large intestine, much of the water and virtually all of the nutrients have been absorbed. Additional water is absorbed in the large intestine, and the solid material is compacted to form fecal waste. Now let's focus on the protein component of the meal as it moves through the alimentary canal. Although protein food particles are mechanically broken up in the mouth, the chemical digestion of proteins does not begin until the food reaches the stomach. After peristalsis propels food particles into the stomach, muscular churning helps break them up. For proteins, however, chemical processing is far more important than mechanical processing. In the stomach wall, there are parietal cells that produce carbonic acid. The canals alongside the parietal cells are continuous with the stomach lumen. The carbonic acid immediately dissociates into a bicarbonate ion and approaches the canals along the wall. There are parietal cells that produce carbonic acid. The canals alongside the parietal cells are continuous with the stomach lumen. The carbonic acid immediately dissociates into a bicarbonate ion and a proton. The protons are actively pumped into the canals, and the chloride ions enter the canals through chloride channels. The protons and chloride ions combine in the canals to form hydrochloric acid, or HCl, a component of gastric juice. Gastric juice is highly acidic, having a pH as low as 1.5. The HCl then enters the lumen of the stomach. The HCl helps break down protein-containing components of the food into individual proteins. The HCl then denatures the proteins, which unfold.
I don't understand that that's a fantastic question. Stop glazing me, bro. Yeah, that's why it's kind of weird that they said that.
And separate into long polypeptides. Mucus lining the gastric epithelium protects the stomach cells from denaturation and from damage by the stomach's own acid. Specialized stomach cells called chief cells secrete the inactive compound pepsinogen, which is converted to the enzyme pepsin by contact with the acidic environment of the stomach. Pepsin breaks peptide bonds, reducing the proteins and long polypeptides to short polypeptides. Protein digestion is completed in the small intestine with the aid of pancreatic enzymes. The enzyme enterokinase, produced in the small intestine, activates the pancreatic enzyme trypsinogen, forming trypsin. Trypsin then activates other protein-digesting pancreatic enzymes. By the time the partly digested food reaches the end of the small intestine, short polypeptides have been broken down into individual amino acids. Cells lining the small intestine absorb amino acids in much the same way as they absorb glucose. The bloodstream then carries the amino acids to individual cells throughout the body. The remaining wastes
So
are
is
eliminated.
amino acid uptake into the small intestine epithelium also it's not active transport. It's just co-transport based on the sodium gradient. And the baso. Okay. Alright, so it's not active transport.
Via the large intestine. Now let's focus on digestion of the fat component of the meal as it travels down the alimentary canal. As the food particles are mechanically broken down in the mouth, cells in the tongue secrete the enzyme lipase. This enzyme begins to process the fats, also called lipids, chemically. However, major chemical digestion of fats does not begin until the food reaches the small intestine.
So we have salivary lipase and amylase, but we don't have like
Pulses propels
salivary
the particles into the
protease.
stomach. Muscular churning helps break them up. Because the fats are insoluble in water, they tend to aggregate into large globules as they're churned. The pancreas secretes several enzymes into the small intestine, including the enzyme pancreatic lipase. Lipase breaks down fats. However, before the molecules in the fat globules can be digested by enzymes, the globules must be broken up or emulsify into globules, and because the fats are insoluble in water, they tend to aggregate into large globules as they're churned. The pancreas secretes several enzymes into the small intestine, including the enzyme pancreatic lipase. Lipase breaks down fats. However, before the molecules in the fat globules can be digested by enzymes, the globules must be broken up or emulsified. Bile, secreted by the liver and stored in the gallbladder.
Oh wow. Okay, so no, so all of the digestion enzymes are I don't know why it said I was a half step away. I feel like I said it exactly right. Wait, can we close this one?
Called bile salts that emulsify fat globules. The gallbladder releases the bile into the small intestine. Fat globules from food, lipase from the pancreas, and bile salts from the liver come together in the small intestine. When bile enters the small intestine, the bile salts emulsify the large fat globules into small fat droplets. Pancreatic lipase breaks up the small fat droplets into monoglycerides and free fatty acids. Once fatty acids have been liberated, it's thought that fatty acid binding proteins bring them through the microvilli of the cells lining the small intestine. There, they're processed into protein-coated complexes and released into the bloodstream for transport to fat storage cells and other tissues throughout the body. Again, the undigested waste is eliminated via the large intestine.
Okay. Which part is not part of the alimentary canal? I would guess salivary glands is not. I would probably say excretion. One advantage of having a tube-like digestive tract is that digestion of all compounds can take place simultaneously down the tract. I mean, I don't know if that follows from it being a tube. All these ones that say one try left, I don't really play. I just let Chet do it. Okay, wait. Let's read it one more time. One advantage of having a tube-like digestive tract is that digestion of all compounds can take place simultaneously down the tract. I feel like it's definitely false. Like a gastrovascular thing can do it all at the same time too. Wait, okay, without even knowing, I'm just gonna say wrong. And I wanna see what it says. Dude, I think it's false. But this question is just worded really poorly. Yeah, I feel like it's worded in a way for you to say false, but whatever. Ooh, the liver. Oh yeah, it's accessory organ, but it still counts as the tract. I don't know. I would say probably it's part of the digestive system, but I don't know if it's part of the. Let's see. What does the question specifically say? Alimentary canal. Let's see. What's the hint? Consider the sequential parts. Yeah, I would probably say false. Yeah. Right. The alimentary canal, mouth, pharynx, esophagus, stomach, small intestine, large intestine, anus. Okay, yeah. So those are accessory. I believe that. That makes sense. Wait, that's wrong. It's a low pH. Mechanical digestion of proteins is more important than chemical. Did it say something weird like that? Protein digestion begins in the small intestine. I thought that actually might be right. Proteins that are consumed in the diet are absorbed as following digestion. This actually might be the right one, because digestion is defined as the hydrolysis, and no, actually this is wrong though. In lecture, he just said that protein digestion is intracellular. I think this thing doesn't know what the hell it's talking about.
So continuing, just immerse your food in this acid. On the and then of all these proteases and peptidases that are breaking things down into smaller fragments. So the smaller fragments are oligopeptides. So polypeptides are large fragments, oligopeptides are small fragments, until you finally get into ditripeptides, so 3 or 2 amino acids, or just 3 amino acids. Okay, so we're whittling around these long chains of amino acids into smaller and smaller and smaller pieces. So from proteins, into polypeptides, and oligopeptides, to tri-amino acids, diamino acids, we call dipeptides and tripeptides, and 3 amino acids. Now the brush border transporters are absorbing these di- and tripeptides and amino acids into the cells.
Chat's so dumb, bruh. So then this is wrong. That's wrong. So then well, it's like, what is the definition of digestion? If it's hydrolysis, then I'm gonna guess this one. This is also wrong, bro. It's got to be this one then. I don't know if I get to try again. Bro, I feel like I'm gonna get this one wrong. What is it? High pH is like definitely wrong, so I feel like I want to do this one. Yeah. It's funny, it just kind of contradicts what he was saying, though. But I mean, a lot of the hydrolysis, though, is happening extracellularly, so it's only like the final bit. Choose the correct statement from the list, but wrong. That's true. Carbohydrates are fully wrong. Bile salts from the gallbladder are essential. That's also wrong. Choose the correct statement from the list below. Free fatty acid absorption occurs in this. That's wrong. Fat digestion begins in the stomach. Fat digestion begins actually in the mouth. Apparently we have lipases in saliva. Bile salts from the pancreas are essential. Yeah, that's true. Wait, what? Oh, I didn't even read it correctly. They try to trick me up, bro. This is not right. Oh, this is the right one then. But with the one try left, I don't play. Why did I skip over this one? That's definitely it. Why did I skip over that one? Yeah, that's definitely it. Bro, so many videos. Holy cow.
The stomach and its secretions. One of the major functions of the stomach is to process food into a liquid suspension. The stomach does this by producing a digestive fluid called gastric juice and mixing it with the food. This figure illustrates how gastric juice is produced. The stomach is a hollow organ, and the space inside this organ is called the lumen. As you can see in this cross-sectional view of the stomach wall, the interior surface of the stomach contains numerous infoldings, or pits, lined by epithelium. The spaces inside the pits are part of the lumen. To understand how gastric juice is produced, let's zoom in on the cells that make up the lining of one of these pits. There are two types of cells that secrete the main components of gastric juice. One type of cell, the chief cell, secretes a protein called pepsinogen into the lumen. The other type of cell that contributes to gastric juice is the parietal cell. It actively transports hydrogen ions into the lumen. Chloride ions then diffuse into the lumen through channels in the parietal cell membrane. Within the lumen, the hydrogen ions and chloride ions combine, forming hydrochloric acid, HCl. How does hydrochloric acid affect the properties of the gastric juice in the lumen? The hydrochloric acid that forms in the lumen makes gastric juice very acidic, giving it a pH of about 2. The low pH of gastric juice aids digestion in several ways. It disrupts the extracellular matrix that binds cells together in meat and plant material. It denatures proteins in food, exposing their peptide bonds for later attack by digestive enzymes. And it converts a few molecules of pepsinogen to an active protein-digesting enzyme called pepsin. Pepsin has an optimal pH that's close to 2, so it works best in the very acidic environment of gastric juice. In addition to beginning the digestion of proteins in food, pepsin converts additional molecules of pepsinogen to pepsin. Those pepsin molecules, in turn, convert remaining pepsinogen molecules to pepsin. This process is an example of positive feedback, because the product of the reaction, pepsin, catalyzes further reaction. The positive feedback loop drives to completion the conversion of pepsinogen to pepsin. What might happen if hydrochloric acid was produced inside the parietal cells instead of in the lumen of the stomach? If hydrochloric acid was produced inside the parietal cells, it would denature proteins inside the cells. But because hydrochloric acid is produced in the lumen instead of in the parietal cells, the proteins inside the parietal cells aren't denatured by the very low pH. However, each parietal cell and chief cell has part of its external surface in contact with the lumen. What protects this surface from being digested by gastric juice? A layer of mucus lining the lumen provides some protection. This protection isn't complete though, so the cells must be continually replaced through cell division. The mucus that lines the lumen is secreted by a third type of cell, the mucus cell. Mucus cells also line the pits. As the cells lining the pits continue their secretions, gastric juice passes from
Bro, if you were a cell, you'd be a mucus cell.
the lumen inside the pits to the main chamber of the stomach. Therefore, the pits function as glands and are called gastric glands. Muscular contractions of the stomach wall mix the gastric juice with food, promoting further breakdown of the suspended food particles. Sphincters at each end of the stomach keep this mixture contained within the stomach. The upper sphincter opens periodically to allow more food to enter from the esophagus. As digestion proceeds, the lower sphincter begins to open rhythmically, allowing the mixture to enter the small intestine one squirt at a time.
Oh, yo, what the fuck, bro? Yeah, that's so funny. Which is the funniest example of positive feedback? Pepsin, well, I mean, that sounds alright. How do hydrogen get into the lumen of the stomach? A researcher added a mixture of animal proteins to a physiological buffer solution in a test tube incubated at 37°C. The researcher then added purified pepsin to the mixture, but even after several hours, the proteins were not digested. Well, the pH was too high. What is one function of mucus in the lumen? It protects the epithelium from being digested by gastric juice. The epithelium that lines the stomach has many deep infoldings or pits. In which way do these pits function as glands? They secrete gastric juice. Whoa, what the hell is this? The structure of fats make them insoluble in water. That was weird. Damn, Riley was here? What the hell? I just been grinding, dude. Okay, that emergency warning, I don't know where that came from. Um, wait, fatty acid. Carboxyl. Alright, this figure shows how fats are absorbed from the lumen of the small intestine and ultimately reach the blood. In small intestine epithelial cells and in lacteals. Chylomicrons leave this structure by exocytosis. This is an epithelial cell. This structure contains lymph. Lipase breaks down triglycerides. Monoglycerides and fatty acids are reassembled to form triglycerides. Triglycerides are coated with phospholipids, cholesterol to form. Remember that molecules that are soluble in water are hydrophilic. Molecules that are not are hydrophobic. During their digestion and absorption in the small intestine, fats are packaged into hydrophilic forms. Place labels under the flowchart indicate whether the bolded structures are hydrophilic or hydrophobic. Fats, hydrophobic. Fatty acids. Fatty acids are probably still hydrophobic, right? Because fatty acids are defined by their carboxyl group, which is a hydrogen bond, but mostly it's nonpolar. It's almost certainly nonpolar, so it's probably and then if that's the case, if we know one of these is hydrophobic, then their monoglyceride is probably hydrophobic too. Although glycerol is pretty polar. Look at all these hydrogen bonds. Triglycerides. Philic is also hydrophobic. Now these are probably hydrophilic. Let me see if I'm right, but yeah, that makes sense. That's actually a good problem because it helps you show that like the whole process is just getting them so that you can transport them in water. Which statement best describes why mechanical digestion is an important digestive process? What is the cecum? I don't even know what the cecum is. The main thing is for herbivores, yeah. Which statement best describes why cells of the digestive system secrete proteolytic enzymes, such as pepsin? In their inactive forms. Oh, in their inactive forms. Different types of food are eaten by various groups of animals, but it is usually true that the teeth of herbivores tend to be sharper than the teeth of carnivores. So animals typically produce Is that true? It's the only one that kind of makes sense. Yeah, it could be. It could be stomach. Intestine's probably right. Which of the following is the fate of organic molecules that are absorbed in All right. Good stuff. I'm gonna make some food, and then I don't know.